All right, so now that we have PopArt installed and open, we need to import two different types of files to make sure we can actually create our haplotype network. So here in the left, I have that PopArt window open. You can also adjust this if you want. You can make it bigger or smaller. If you look here on the left-hand panel on our data viewer, right now it's empty. We haven't added anything, but you can see there are two different tabs, one for alignment and one for traits. And so those are the two different types of files we need to import in order to create a haplotype network. So let's start with the alignment, and both the alignment and the traits file are available on Canvas on the same page that you got the full written instructions from the lab for. So just a brief introduction to alignment files. It has a .nex file extension. It's called a Nexus alignment file. And really what this file is showing you is the particular sequence of that MYB12 transcription factor that we're comparing across every single individual. Remember, we had 396 plants total in this data set, so each one of those plants has a line in this Nexus file alignment. The part on the left, so there's basically two different columns. The column on the left is the sample name for that particular individual. So NF1, for instance, is from that NF, Nacimiento Ferguson. Is from this Nacimiento Ferguson location, and that's the first plant that was there. So that's what the information in the left column is, and the information in the right column is that sequence information for each one of those 396 flowers. So that's what's in our alignment file. You don't need to open the alignment file before you import it into PopArt. In fact, I think I recommend not doing that just because there are some chances that formatting might get a little bit funky. Also, your computer might not know what program to use to open this type of file. So you don't need to open this on your computer. I just wanted to show you briefly what it looks like so you have a basic understanding of what we're doing here. So the alignment file is basically that data that's showing us the DNA sequence information for each individual in our data set. So if we go back to PopArt, okay, I tried to adjust the window. Hopefully you can see the top now. So if we go to this main part of navigation panel for PopArt, if you go File to Import, we want to import an alignment. Change this file type to all files because remember it's a .nex, so it won't show up unless you change that. And navigate to wherever you saved that alignment file that you downloaded from Canvas. I recommend setting up a separate folder for our lab. So I will find, where did it go? Okay, so MIB12.nex, that's what we want to import. So you can either double click on that or click on it and click OK. And now if you look in this alignment portion of our data viewer, we have all our data there. So all of those 396 individuals, each one has the sequence information there. So that's the first step. The next thing we need to do is add in a traits file. And this is very similar to what we did before in DNA Subway when we were adding in a metadata file. That metadata file is what told the program which of these different samples belonged with which sampling group. That's very, very similar to what a traits file is. Again, just to show you a little bit about what these types of files are, this is a traits file that I created for our data set. As I mentioned in the lab instructions, this is rather tedious and I didn't think it was the best use of your time. So if you have questions about how I did it, I'm happy to answer them for you, but I tried to do that legwork just because I don't think it's a good use of your time. But overall, what these type of traits files look like is there's a top header file that has information for all of the different possible sampling sites or different populations in this data set. So there were 9 different populations total, so there are 9 different names here at the top of the traits file. And then at the bottom of the file here, just like before in that alignment file, we have one line for each individual that's in our data set. And this right here, NF1, that name has to match exactly the same name that was used in the alignment file. So again, it needs to be able to match up. So each individual has its own line, and then what's shown here to the right, these series of binary 1 and 0 numbers, this is how we tell PopArt which population this NF1 individual is from. And the way you do that is you put a 1 in the slot that corresponds to the population in your header that this individual is from. So if we look at these numbers here, there are basically 9 different slots. The first slot has a 1 in it, and if we look up here in the header file, the population that corresponds to that first spot is Nacimiento-Ferguson. That means that this particular individual was from Nacimiento-Ferguson. That's how we tell PopArt where these different individuals are from. If we scroll down to the BC2, for instance, Big Creek is our second possible population site. So now for BC2, we have a 1 in that second position because that corresponds to the population that we want to tell PopArt this sample is from. So that's overall what a traits file looks like. Again, you don't need to open it or do anything with it. You can just download it from Canvas as I provided it to you. I just wanted to give you a bit of background about what these files look like. So let's go back into PopArt now, and we go to that same file to import, and now we want to import a traits file. So go to wherever you downloaded that file. Here is mine. So like I said before, you can just double click on it, or you can click on the file and then click open. Either way is fine. We do not want to clear the alignment data. Like I said, we want to match up our alignment data with our traits file, so we don't want to clear it, we want to keep it. So no for clear alignment data. And then here, you shouldn't have to change anything. The delimiter should be set to comma. Table has header rows. We want it to recognize that header of populations we have. And you can do a quick check here. All those NF individuals are from Nacimiento-Ferguson. They should have a 1 in that column. And if we look here, that matches up. See, if we take this off, now it's messed up. So you shouldn't have to change any defaults, but make sure comma is selected and table has header rows. Just as a quick note, there's something funky going on here at the beginning of the Nacimiento-Ferguson. I'm going to try to figure out how to fix that before I give you the traits and alignment file. But if I can't figure it out and you see this in your data, that's fine. It's just some kind of weird formatting thing. So once we have comma checked and table has header rows, what you do is click OK. And so now what we have in this left data viewer panel is it tells us how many samples we have for each one of those populations. Now we have our traits filled in and our alignment. And so if you go to network now, it gives you the option to make these different networks. So I'll cover that in the next video, but now you should have an understanding of how to import in a traits and alignment file into PopArt, which will then allow you to make that haplotype network.